Thank you very much. I'm actually still currently in Cambridge, but will shortly be moving to Sheffield. So I'd like to thank the organiser for his very kind invitation. I hope no one's going to be too disappointed that I'm not really talking about adaptation to global change, and I'm certainly not talking about anything in the Mediterranean. So I'm going to be talking about these butterflies, which are Heliconeus butterflies, and are found in the neotropics, so South and Central America. But I am going to be talking about hybridization and species boundaries. So this is quite a nice system for looking at these types of questions. So for those of you who are not familiar with the Heliconeus butterflies, they are a radiation of about 40 species that's radiated over about the past 10 million years or so. And even within species, there is quite a lot of diversity. So this shows Heliconeus melpomene, and these are different forms within this single species that occur in different geographic regions in South America. And this is its sister species, Heliconeus sydno. And this is one of the things that Heliconeus is most well known for, which is mimicry. And this is Heliconeus erato, which is its co-mimic, but quite distantly related. And again has these different colour patterns, and in each geographic region, there's a co-mimic between each form of Heliconeus erato and Heliconeus melpomene. And again, a co-mimic, a distantly related co-mimic with Heliconeus sydno. So we have these different levels of divergence within Heliconeus melpomene. We've got different racial forms that aren't true species yet, but are possibly on the way to speciation. We then have sister species that seem to be good species. They very rarely hybridize, particularly in the wild, although rare hybrids are found. So we know there is some level of gene flow between these sister taxa. So this is, for example, between Heliconeus melpomene and the sister taxon, which includes Heliconeus sydno and Heliconeus timoretta. And one of the interesting things that you'll see here is that Heliconeus timoretta actually shares some of the same colour patterns that Heliconeus melpomene does. So this kind of raises the question of, have these colour pattern alleles been shared between these sister species because of this very occasional gene flow between the species. And then we can look at slightly more distantly related taxa. So in this case, Heliconeus melpomene, and this group here, which are called the sylvaniforms. And most of these have these sort of striped colour patterns that look very different from Heliconeus melpomene. But again, even rarer occasional hybrids are found in the wild, and under extreme conditions, they occasionally hybridize in captivity. And again, we find one species, so this is Heliconeus elevatus, which is within this clade, but has a melpomene-like colour pattern. So again, raising the possibility that even these more distantly related species might be sharing colour patterns by hybridization. So this raises some questions, some of which I'm going to talk about. So what are the genome-wide patterns of divergence that we see at these different levels of divergence? And what are the levels of gene flow between these? Is there evidence at a genomic level for gene flow between these distinct species? If so, how extensive is this? And when did it occur? Has it just been a single burst of hybridization at some point in the past? Or is there sort of ongoing gene flow between species? And finally, have these colour pattern alleles been shared between species, which would suggest that this hybridization can sometimes lead to adaptive consequences? So starting off at the least divergent level, so these are different colour pattern forms of Heliconeus melpomene that occur in Peru. And within the hybrid zone, we find admixed phenotypes, so suggesting that there's hybridization going on. And from what we know about the genetics of the colour patterns, it seems like mating within the hybrid zone is essentially random. So it seems like there should be a lot of gene flow going on between these different subspecies. But is this what we see at the genomic level? The simple answer is yes. So this is using RAD data, so restriction-associated DNA sequencing. For those of you who don't know what that is, basically you chop up the genome with a restriction enzyme and then you sequence just from those restriction enzyme cut sites. So it's a way of subsampling the genome. In this case, we're sequencing about 5% of the entire genome. And we've done this for 30 individuals from 2 species, so the co-mimics, Heliconius melpomene and Heliconius aurato, across their hybrid zones in Peru. And we've done this from pure populations of both of the subspecies, and then from 10 individuals that are of a hybrid phenotype from the center of the hybrid zone. And this shows a structure analysis. So here we assume that there are 2 populations that are contributing to this genetic structure. And each of the horizontal bars shows the contribution of each of those 2 populations to that individual's genome. So, for example, in melpomene, we can see that most of the individuals have sort of a 75-25% split between these 2 population types, and then there are a few individuals that are pure for one or the other. But the main thing to notice is that this doesn't at all reflect the subspecies distribution. So, suggesting that these 2 color pattern subspecies types are essentially the same thing. And we see a similar thing with a principal components analysis of the genetic variation in this population. So these are the same outlier individuals that are being picked up by the structure analysis, and most of the individuals look essentially the same, with no structuring by color pattern. With Heliconius aurato, the structure analysis picks up even less genetic structure within the populations, and the principal components analysis, we can see there is some splitting up along this principal component 2 by color pattern type, but there's sort of continuous variation, and this is less than 5% of the genomic variation in total. So there's very little variation that's being partitioned by these subspecies. And we can also see this with FST. So this is just considering these pure populations and the genetic differentiation between them. And in general, there's very little genetic differentiation and just a few sites across the entire genome that are showing much stronger differentiation. And these are the sites that we're really interested in terms of the ones that are going to be determining this phenotypic differences that we see. So we can see where these are on the genome. And this now is just looking at Heliconius melpomene. So this bottom plot shows the FST values and where they are on the genome. And the ones in red are those that show evidence for being significantly higher than what we would expect under neutrality. So these are the ones that seem to be under selection. And you can see they're strongly clustered in the genome in basically these 3 locations. This top plot shows an association analysis looking at associations between SNPs in the genome and the color pattern variation. And what we can see is there are 2 major loci that are controlling most of the color pattern variation that we see. One of them that's controlling most of the red color pattern elements, as well as the shape of the forewing band, which is this locus here, and one that's controlling the yellow color pattern elements, so the presence of yellow in the forewing and in the hindwing, and that's located here. And most of the strongly differentiated loci that we see are corresponding to these loci that are controlling color pattern variation. There is one other interesting region that we find at the end of chromosome 6 here that doesn't seem to be controlling color pattern variation, but is strongly differentiated. So this could be a candidate for something that's possibly involved in, for example, altitudinal variation. So this cline that we see in color pattern also occurs across an altitudinal gradient. So these could have be important for other aspects of ecological adaptation in these butterflies. But the basic pattern that we see is that between these subspecies, there's very low genetic differentiation, so high gene flow, and just a few loci that are strongly differentiated, driven by differential selection in the 2 populations. So we can then move on to looking at a more distant comparison, so between sister taxa, in this case Heliconius melpomene, and its sister taxa Heliconius sydno and Heliconius timoretta. And this is again using the same type of RAD data. And this is a phylogeny created with that RAD data, showing that these taxa are clearly distinct and monophyletic with respect to each other. This is a similar type of structure analysis to the one I showed before on the individual populations, this time across different species. And in this case, there are 5 different populations that have been inferred to be present. So 3 of these are present just within Heliconius melpomene. So this yellow one are Heliconius melpomene populations that are occurring west of the Andes. This red block are populations that are occurring east of the Andes. And then we also see a split between Heliconius timoretta, which is also occurring east of the Andes, and Heliconius sydno, which is occurring west of the Andes. And the interesting thing, I'm not sure if you can see it, but hopefully you can, is that between the populations that are occurring west of the Andes, we see some gene flow, so there's a little bit of pink here, and a little bit of red in the pink there. And we also see some evidence for some sharing of alleles between the west of the Andes populations. So there's a tiny bit of green here that you can maybe see, and a tiny bit of yellow down here. So we want to see if we could test this further. So under this scenario, we would expect that regions of the genome that show strong associations with the geography would be indications of bits of the genome that are flowing between species, particularly if they show stronger associations with geography than they do with these 2 major taxon. So, for example, things that are shared and similar between Melpomene and Sydno in Panama, but distinct between, as compared to populations of Melpomene and Timoretta in Peru, would be evidence for things that are being shared locally in these different populations. So this is what I did. This is looking using analysis of molecular variants at individual RAD loci, and grouping populations either based on which of these 2 taxonomic groups they belong to, or by which of the geographic regions they were found in. So you can see that parts of the genome, as we would expect, are splitting up by these taxonomic groups. This is, of course, what we expect, because we can see from the phylogeny that these are distinct. But we also find a large number of these RAD loci are being separated by the geographic population that they occur in, irrespective of which taxonomic group they occur in. So they're showing stronger associations with geography than they are with taxon. And in total, I found 123 of these loci that were significantly associated with geography, but not with taxon. And based on the number of loci that we had and the probability thresholds, the null expectation would be that there would be 39 of these. So we're finding more than we would expect just under chance. And if we look at where these are distributed in the genome, so in this case we've got anything that's going below 0 shows a stronger association with geography, and anything going above 0 shows a stronger association with taxon. You can see that the points that are going below 0 are essentially spread out across the whole genome. So bits of the genome that are sort of dispersed across the whole genome are being shared in distinct geographic populations. And this can't really be explained by ancestral variation that has sorted into the distinct species, because then we wouldn't expect it to be distinct between the geographic locations. But we wanted to look at this in more detail and really try and quantify how much of the genome is being shared between different species, and also when did this occur. So this was work that has mostly been done by Simon Martin, who's a PhD student in the group. So he's been using these that are tests that have come to be known as ABBA-BABA tests, So this is essentially a way of quantifying the amount of shared derived variation between two populations that we think might be hybridizing. So in this case, sympatric populations of Heliconius timoretta and Heliconius melpomene amaryllis. So we look for sites that are shared with these, between these two taxa, but divergent in an outgroup and also in one of these in-group Heliconius melpomene species. And compare this to what we would expect just under neutrality, which is essentially where the BABA bit comes in. So this can be done using any one of these different melpomene populations that were sampled here, and this is now using whole genome data. And depending on which of these populations we use, it will tell us something different about which part of this ancestral history of divergence, when did the gene flow happen? So if we look, for example, just using this very closely related population here, we will only be able to detect gene flow that's happened very recently. Whereas if we use a more distantly related Heliconius melpomene group, for example, Rosina out here, then that can tell us about gene flow that's happened right back to the common ancestor of those two melpomene populations. And we can also use these ABO-BABA tests to estimate the fraction of the genome that's been shared between the species. So when we do this, if we use this closely related population, where we're just looking at this most recent amount of gene flow, then the estimate is quite small. It's just a few % of the genome is estimated as being shared. Whereas if we use this more distantly related population of melpomene, then the estimate of what's being shared is almost at 15% of the genome being shared between species. So what this indicates is that gene flow has been ongoing over much of the history of divergence. So it hasn't. So if it had just all been very recent gene flow, then we'd expect this estimate to be the same as this estimate. But it indicates that in fact gene flow has happened over a large portion of the divergent history of the species. And we can also see that it's much higher when we look at the whole genome compared to when we look at the Z chromosome. So it looks like there are some bits of the genome that are not being shared as much as others. And in particular, we know that the Z chromosome can harbour sterility genes in the system. So this kind of fits with what we know about the biology of the system. But we can also test this idea further, that there have been differences in gene flow over time, and in particular that the idea that there has been some recent gene flow by looking at the level of linkage disequilibrium that we see at these different gene flow levels. So for things that are being shared very recently, we would expect there to be much larger blocks of linkage disequilibrium. So we can test whether this really is the case. And in fact, it is what we see. So this shows the level of this shows the distance between the SNPs, so how close they are physically in the genome, and this shows the correlation between them, so the degree of linkage between them. And this top line is for these SNPs here, which are the ones that we're detecting here, so the things that seem to have been recently shared between species. And what we see is that the linkage between these very recently shared SNPs decays much more slowly. So supporting this idea that there has been some very recent gene flow between species leading to these big linkage blocks, but also that there has been this more ancient stuff, which is what we're picking up when we look at these older comparisons. For those of you who've been paying attention, you may also have noticed that these two species that I've been talking about also share the same color pattern. So this is Helicones timoretta that occurs in Peru, and this is Helicones melpomene amaryllis. So these are also candidates for species that may have shared the color pattern alleles between them. So we wanted to also test this further. And this is work that I did in collaboration with Kanchon Desmahapatra, who's now in York. So this is now using targeted sequencing, focusing on one of these color pattern controlling regions. What I'm showing here is the region controlling the red color pattern variation. And this, at the moment, is just showing divergence between these subspecies. So it's basically zooming in on that, in essence, zooming in on that plot I showed at the beginning of this peak of FST divergence around the red color pattern gene, but now looking at much finer detail. And this allows us to pick out where we think the functional region controlling red color pattern variation is. So we can really see in quite a lot of detail where the strongest divergent peak is in this comparison. So going from here, we can then compare, look at what's happening from a phylogenetic perspective when we look at these different species that share different color patterns, but belong to different taxa. So this is Helicones melpomene again at the top, showing these two different color pattern forms. This is Helicones timoretta. So here we have the Peru population that I showed that shares the color pattern with this Peru population. We also have a rayed Helicones timoretta. This is found in Colombia. And these two rayed color pattern forms overlap in their distribution as well. And then this is our outgroup, so the sylvaniforms. And again, this one rayed sylvaniform species, Helicones elevatus. And when we look at the sort of outside of where we think the functional regions are in this color pattern controlling sort of block, we find basically the background phylogeny that we find at neutral loci where we have grouping of the species, where these as the outgroups. If we then look sort of within where we find strong associations, but not right where we think the functional region is, we see a jump in the relationships between the species. So we now have these sort of stripy things grouping together, and we have the rayed things grouping together, irrespective of the species. When we then look right where we think the functional region is, we even get Helicones elevatus, so this distantly related rayed butterfly grouping with these other rayed butterflies. So it really does look like there has been sharing of these color pattern alleles between butterfly species, and even between quite distantly related species. And this doesn't seem to be explainable by, certainly not by recurrent mutation, because we wouldn't expect this to influence as many sites as we would need to get with these 50 kilobase windows in order to produce this jumping of phylogenies. It also seems very unlikely that it's a shared ancestral polymorphism, because again, we would expect there to have been enough time for recombination to break down these big blocks. So we wouldn't see these big 50 kilobase windows producing these phylogenies where we have convergent color patterns grouping monophyletically. So overall, just in summary, in these butterflies, we see very little population structure across hybrid zones between subspecies types. And it's only really the color pattern loci and possibly a few other divergently selected loci that are really strongly differentiated. Moving to more distantly related species, there's evidence for gene flow between these sister species, and evidence that this has been ongoing over much of their divergent history, and also that it's going on across much of their geographical range. It seems that up to about 15% of the genome is being shared between some of these sister species pairs. And also, as I said, this has been going on over much of their divergent history. And finally, that there is evidence that some of this gene flow can be adaptive. So in particular, these color pattern loci seem to be being shared between sister species, and this has likely allowed the species to come into the same mimicry rings and therefore share protection from predators in that area. So finally, it just remains for me to thank a few people. So this work has mostly been carried out in the butterfly genetics group in Cambridge, where Chris Jiggins is the PI of the lab. The hybrid zone study that I spoke about has been in collaboration with a group at the University of Puerto Rico. In particular, I've been working with a postdoc, Maite Ruiz. And then I mentioned Simon and Kanchan already, and these are some other people that have been involved. And thank you very much for your attention.